Hello viewers, this is Abdul Rahman and you are watching Student's Diary. Our today's tutorial is about working principle and importance of G-protein coupled receptor. When a signal or ligand which carrying information bind to specific G-protein coupled receptor, it result in activation of G-protein so that the G-protein relay message downstream inside the cell through production of large number of second messengers, which could be cyclic AMP, IP3 and diacylglycerol to regulate multiple body functions that could be growth, taste or smell or any other sensation or endocrine response and much more. Let's see how the G-protein coupled receptors look like and how it work in inactive form. We assume this is a cell membrane. Inside the cell is a nucleus and intracellular environment, while this is the extracellular part. This is how these receptors look like. It has ligand binding site outside the cell, while it span around the membrane 7 times. Remember, this whole receptor is made up of single polypeptide. In inactive form, the alpha subunit of G proteins are attached to the receptor. That's why it's called as the whole assembly is known as G protein coupled receptor. Of course, there are some other subunit that is called as alpha and beta subunits. There might be many other signal in extracellular environment, but if not specific receptor, then it will not bind to it. During inactive form or unavailability of receptor specific ligand or signal, GDP or guanosine diphosphate will remain attached to alpha subunit to keep it in inactive form. As soon as suitable ligand bound to these receptor, GDP will be replaced by GTP, that is guanosine triphosphate, which will result in activation of G protein. This G protein along with other subunit will detach from receptor and will transfer the message to adenylyl cyclase, which is also attached to the cell membrane. Adenylyl cyclase will convert AMP, that is adenosine monophosphate, to CAMP, that is cyclic AMP, which is a second messenger. Similarly, this G protein may also activate phospholipase A2 depending upon the type of information it have. Phospholipase A2 will convert phosphatidylinositol into two second messengers, that is IP3, which we called as inositol triphosphate, and DAG, which is also called known as diacylglycerol. These second messenger will amplify and relay message to target organelles or nucleus to perform the activity, which could be either gene regulation, transcription or any other task. As for the ligand remain bound to the receptor, it will be in active form relaying message to the target organelles or nucleus. But if the half-life of ligand completed or no more signal available to receptor, the GTP will be again replaced by GDP along with the movement of G protein subunit towards the receptor to switch G protein coupled receptor to inactive form, so that cycle continue like switch on and switch off mechanism depending on the availability of ligand. At the end, let's have a look on the importance of G protein coupled receptor. It is the most diverse group of extracellular membrane receptor and present almost in all eukaryotes animal, either it's plants, fungi or yeast. Interestingly, there are almost more than 1000 G protein coupled receptor only in humans, and each of these receptors are specific to ligands or signal. These signal could be in the form of light energy, some kind of taste or peptides, proteins or even lipids. G protein coupled receptor is the target of most of the medicine available in the market and also the target for the modern research. Each G protein coupled receptor consists of single polypeptide, as I have mentioned earlier, and which spans 7 times around the membrane. That's why sometimes it's called as 7 transmembrane receptor. That was all for today. For more tutorial, please don't forget to subscribe and press the bell button so that you get notification timely.